Welcome everyone to our video on conjugation. So conjugation is a form of horizontal gene transfer that requires direct contact between two cells, like you see here. A common example involves the F plasmid, which stands for the fertility plasmid, and you see that here in pink. This small circular piece of DNA carries the genes needed for conjugation, including genes involved in producing the pilus, which is the structure that is going to be directly involved in contacting that neighboring cell and then ultimately allowing the transfer of the DNA. So, real quick, I want you to pause this video and think about what are the features of a plasmid? What is a plasmid? Why might this process be beneficial to the recipient cell? Which, if you look closely, the recipient cell is an F minus cell, meaning it does not have the F plasmid. Okay, so the cell with the F plasmid is referred to as the F plus. It is the donor cell. It is going to initiate this process of conjugation and ultimately share that plasmid. Okay, so at the end of the day, they'll both have it. They'll both be F plus. So pause this video and answer that question. Jot it down on your notes. All right, so remember the F plasmid is separate from the bacteria's chromosome. Okay, so the chromosome is in blue, the plasmid is in pink. And so the plasmid, notice that it looks similar to the chromosome. It is also a double stranded circular molecule, but again, it is extra chromosomal. That means it is not part of the chromosome. That means it is not essential for life. Okay, all of the DNA that is essential is part of the chromosome. If the chromosome becomes damaged or somehow was not present, that cell would not be able to survive. But should the bacterium lose a plasmid, that's okay, because remember, the plasmid is just a bonus feature. It offers some sort of advantage to the cell, like maybe antibiotic resistance. All right, so while I like this picture, it does kind of miss the integral first step, and that is that our F plus cell, and I'm not going to draw, I'm not going to draw everything with the F plus cell. But what I do want to point out is that the F plus cell is the one that initiates this process. So the F minus cell was over here minding its business, and the F plus cell is like, hey, you don't have this fertility plasmid. Well, let me hook you up. And it starts building and extending the pilus out until it latches on to this neighboring recipient cell. And so I think of the pilus almost like a fishing line where it is built and extends outward, like you're casting a fishing line. It attaches to that recipient cell, but then it reels it in and it brings the cell closer. So I'm just going to try to show it coming in closer. Okay, now it's not, of course, changing side or anything. Okay, but it's reeling that cell in until it is close enough, like what you see in the picture here. And that's where it's actually going to form a more stable connection that we call a mating bridge between them. Okay. So when the cells are this close right here, y'all, we refer to that as a mating bridge. Okay. So now take a look at the plasmid itself and think about the little reminders that I gave you earlier about plasmids. And I want you to notice that the plasmid itself is a double stranded molecule. Okay. But the donor, excuse me, the donor cell doesn't send the entire plasmid into the recipient all at once. Instead, one strand of the F plasmid is cut, like you see in the image, and it's fed through that pilus. And what's going to happen is a copy of that single strand that's going to get transferred through the mating bridge is going to undergo DNA replication. In fact, that's going to happen on both sides, right? Because ultimately, the recipient cell is going to get a single copy of it. And then, if it just stopped there, both cells would have a single strand of a plasmid, but a plasmid is a double stranded DNA molecule. So DNA replication is going to occur in both cells. Okay, the donor replaces the strand that was originally transferred, so it can have a complete double stranded F plasmid. And then inside the recipient cell, that transferred strand is acting as a template for building the complementary strand. So at the end of the day, the recipient has its own complete double stranded copy of the F plasmid. Okay, so remember, the original donor was F plus, it had that fertility plasmid, and it remains F plus because it still has a complete intact copy of that plasmid. But now the F minus recipient has the fertility plasmid. It is also F plus. So at the end of conjugation, both cells contain a complete F plasmid and both now have the potential to transfer it to other bacteria. Okay, so this is going to explain how plasmid carried traits, like things with antibiotic resistance, can spread rapidly through a bacterial population. Now, what I do want to mention is sometimes a bacteria has multiple plasmids, or through a phenomenon that you do not need to worry about how it happens, but the F plasmid can actually combine with an R plasmid, which is what we call the resistance plasmid. And so sometimes the cell can actually have like this super snazzy plasmid that codes for both the pilus and antibiotic resistance. And so conjugation is able to spread more than just the F plasmid and do more than just give a recipient cell the ability to form a pilus. Because if there's more than one plasmid, or if there's kind of like this super, you know, crazy cool other plasmid that it has, then it is able to share that as well. All right, y'all. I know this one is pretty snazzy. Let me know if you have any questions. Bye, y'all.